Hello everyone, welcome to Get My Class. So let's read the question here. The question says, what are the basic features of expression vectors? Option A, ORI, selectable markers, MCS, promoter, transcription initiation site, translation initiation site. Option B, ORI, selectable markers, MCS, RBS, transcription initiation site. Then option C, selectable marker, MCS, transcription initiation site, translation initiation site. Option D, ORI, selectable marker, MCS, promoter, RBS, transcription initiation site, translation initiation site. Now the question says, what are the basic features of expression vectors? So we have to say, or we have to look for the basic features of expression vectors from the options. But before that, we have to understand what are expression vectors. So what do you mean by expression vectors? Okay, expression vectors are the vectors. We know vectors are nothing but the carriers, right? These are the carriers of the gene, right? These are the carriers of gene. Okay, genes, that is the desirable gene, and they will carry the desirable gene and transfer it into the host. Okay, now these expression vectors are actually, they are specific to particular host. That means they bring about, they are very specific towards the expression. Expression in the sense, we are talking about transcription and translation. So whenever we say expression of gene, we are talking about actually transcription, okay, transcription and translation. Transcription and translation. So it will be having all the important components which is necessary for transcription and translation process. Okay. So remember, whenever we say that this gene has been expressed in this particular organism. So what do you mean by expression? Expression, that means that gene can undergo transcription and then further that will give rise to mRNA. Further, mRNA will code for protein. So formation of protein is nothing but expression of that gene. So if you have introduced any foreign DNA, that has to result in the final product, that is the protein. Only then we will say it has been expressed. Okay. So these expression vectors are specific with respect to the type of host in which they will be used, and that will have maximum expression since they are specific with respect to the host, and also they should have certain qualities for being expression vectors. They should have ORI, that is origin of replication. Okay, and they should have selectable markers. Selectable markers like any antibiotic resistant gene, or which will help in finding out the transformants. Okay, this will help in detecting the transformants. Transformants means the ones which have received or where the gene has been expressed, our desired gene has been expressed. Those are called as transformants. So that will be helpful if the vectors have already selectable markers gene. Okay, that is selectable markers or selectable marker genes. Okay, so apart from that, they should have multiple cloning sites. Okay, MCS is multiple cloning sites. Multiple cloning sites. Okay, what is this multiple cloning site? These are the sites where we can introduce, any number of, depending upon how many sites they have, so many different foreign genes can be introduced. That is what is multiple cloning site. Okay, they should be having multiple cloning site. They should also have ribosomal binding site, that is RBS. Okay, ribosomal binding site, because for transcription and translation, and especially for translation, what you need ribosome, right? Ribosome have to, they are the one which will produce proteins, right? Okay, synthesis of protein. So ribosomal binding site also is necessary. Ribosomal binding site. Binding site. Okay, then so that is about translation. They should have transcriptional initiation site, that is the initiation for transcription as well as the initiation of translation. So they should have initiation site for what? For transcription and translation. Transcription and also translation. Okay, so these are the basic requirement of any expression vector. So let us see. Option A says ORI, selectable marker, correct. MCS, correct. Yeah, promoters, of course. Promoters also very important because they promote the entire process. Okay. promoters. That is also necessary. Okay, so promoters, very good. Then transcription initiation site, yes, and translation initiation site, yes. These all should be there in a vector. But what is missing? What is missing? The ribosomal binding site is missing here, right? Ribosomal binding site is missing. So we will not consider this. Let us see all the options and then decide the best option. So sometimes you get questions like this where you all the answers are correct, but you have to choose the correct, the best answer. Okay, option B says ORI, that is origin of replication, selectable markers, MCS, that is multiple chlorine site, ribosomal binding site, transcription initiation. Yes, these also should be there. Then option C says selectable markers, MCS, transcription initiation site, translation initiation site. Yes, that is also correct. Option D says ORI, selectable markers, multiple chlorine sites, promoters, RBS, transcription initiation site, and translation initiation site. So this has the maximum characteristics of for a expression vector. Okay, and hence this should be our correct answer because here all the important characteristics have been mentioned. Whereas in the other option, one or the other characteristics have been missing. So the correct answer should be option D. Okay, so let us conclude the answer. So what is the correct answer? The correct answer is option D. I hope you understood the explanation. Thank you, everyone.